Greetings, I'm Dr. Karl Nath, the Editor-in-Chief of Mayo Clinic Proceedings, and I am pleased to welcome you to the multimedia summary for the journal's July 2020 issue. There are 4 articles that have been selected as our Editor's Choice or Highlights articles this month. Our Editor's Choice this month contains 2 companion review papers on COVID-19, which in concert summarize key epidemiologic and clinical aspects of this disease, outline an approach to management, and provide an overview of drugs and other approaches currently considered and/or investigated for their therapeutic efficacy. The first one is entitled A Collaborative Multidisciplinary Approach to the Management of Coronavirus Disease 19 in the Hospital Setting. This review is authored by Dr. Raymond Razanobly and colleagues from Mayo Clinic in Rochester, Minnesota. The second one is entitled Treatment Considerations for COVID-19, a critical review of the evidence or lack thereof. It is authored by Dr. Prakhar Vijayvargiya and colleagues from Mayo Clinic in Rochester, Minnesota. Beginning with an overview of the essential features of SARS-CoV-2, how the virus infects the cells and how it spreads, Razanobly et al. Broadly outlined the consequences of such infection that range from an asymptomatic state in relatively large numbers of individuals to the spectrum of symptoms observed in COVID-19. Approximately 20% of symptomatic patients develop moderate to severe pneumonia and require hospitalization. Along with commonly observed laboratory findings, the initial patient evaluation is reviewed, and this is followed by a discussion of the diagnostic role of PCR testing, the significance of serologic findings, and the radiologic features observed in COVID-19. The basic management involves antipyretics, analgesia, attention to fluid and electrolyte balance, and oxygen supplementation. Razanobly et al. Emphasized the importance of a team-based approach in managing hospitalized COVID-19 patients and sequentially address major complications and how they are broadly managed, including the hyperinflammatory syndrome, respiratory failure, cardiovascular complications, hepatic dysfunction, acute kidney injury and other renal complications, and the propensity to venous thrombotic disease and disseminated intravascular coagulation. The need to consider the possibility of coexisting infection with other pathogens is emphasized, and isolation procedures are broadly reviewed, including settings when aerosol-generating procedures are performed. The companion paper by Vijay Vigaria et al. Discusses repurposed drugs with antiviral properties, for example, chloroquine, hydroxychloroquine, novel antiviral compounds, for example, remdesivir, immunomodulatory drugs, for example, inhibitors of IL-6 and GM-CSF, and the use of convalescent plasma and neutralizing antibodies. As underscored by Vijay Vigaria, none of these agents have proven efficacy in the management of COVID-19 as of this writing, and they should be employed as part of institutionally approved clinical trials. The authors provide an algorithm for the management of COVID-19 depending upon the severity of the illness. In general, supportive care and close clinical monitoring are provided for asymptomatic individuals or those with mild illness, while for moderate to severe illness, enrollment in an approved clinical trial with specific agents may be considered. If ineligible for such trials, consideration may be given to the United States FDA emergency use authorization of hydroxychloroquine or chloroquine. The paper by Razanobly et al. Returns to the patient recovering from a moderate to severe illness and broadly outlines criteria to be met for appropriate hospital dismissal, requirements for home quarantine, and the importance of telemedicine and remote monitoring in the follow-up of these patients. These 2 companion papers by Razanobly et al. Provide an invaluable up-to-date synthesis of key considerations in COVID-19 and its management, recognizing, however, that the latter will likely evolve as understanding of COVID-19 advances and current therapeutic questions are resolved. Our first highlight article is an original article entitled Marked Upregulation of ACE2 in the Hearts of Patients with Obstructive Hypertrophic Cardiomyopathy. Implications for SARS-CoV-2 mediated COVID-19. This article is authored by Dr. Martin Boss and colleagues from Mayo Clinic, Rochester, Minnesota, and other institutions in the United States. Hypertrophic cardiomyopathy is a relatively prevalent condition afflicting approximately 0.2% of the population and commonly has its origins in genetic abnormalities. For the majority of individuals afflicted by hypertrophic cardiomyopathy, the course is relatively benign and indeed may be asymptomatic, while for a small number there is the risk of sudden cardiac death and heart failure. In the present issue of Mayo Clinic Proceedings, Boss et al. Analyzed the transcriptome in patients with hypertrophic cardiomyopathy and controls, and found that 22% of the transcriptome was differentially expressed in these two groups. Among genotypic groups of hypertrophic cardiomyopathy, just 5% of the transcriptomic changes differ within the three largest genotypic subsets. A striking finding was that the most upregulated gene in hypertrophic cardiomyopathy was the gene encoding for the angiotensin-converting enzyme 2, ACE2 protein, a finding confirmed by RT-PCR and corroborated by increased ACE2 protein expression. These latter findings are quite fascinating given the current interest in ACE2, as this protein, when bound to cell membranes, serves as a receptor for the cellular entry of SARS-CoV-2, thereby facilitating the disease that may ensue, namely COVID-19. ACE2 has distinct enzymatic actions as compared to ACE, the enzyme that cleaves angiotensin 1 to produce angiotensin 2, a vasoconstrictive, hypertrophy-inducing, pro-inflammatory, pro-fibrotic, and aldosterone-stimulating peptide. In contrast, ACE2 exerts countervailing effects because ACE2 converts angiotensin 1 and angiotensin 2 to peptides with actions that are, in the main, vasorelaxant, anti-inflammatory, and antifibrotic. Boss et al. Thus offer the plausible explanation that the markedly increased expression of ACE2 in the heart in hypertrophic cardiomyopathy reflects an adaptive response that may mitigate cellular processes that drive hypertrophic cardiomyopathy. This raises the exciting question regarding the extent to which such upregulation in ACE2 may also be observed in other cardiomyopathies. An additional speculation offered by Boss et al. Is how these findings may relate to the current COVID-19 pandemic. Because of its role as the receptor for viral entry, increased expression of ACE2 may predispose to infection with SARS-CoV-2 and thus COVID-19. And germane to this consideration is that cardiac disease is considered a risk factor for COVID-19. Interestingly, when cells are infected with SARS-CoV-2, expression of ACE2 decreases. Such decrement in ACE2 expression reduces the countervailing salutary actions this enzyme offers, while allowing greater activation of the angiotensin 2 type 1 receptor because of higher levels of angiotensin 2. Boss et al. Thus suggests the possibility that beneficial effects in COVID-19 may be accomplished by either blocking the angiotensin 2 type 1 receptor or complexing SARS-CoV-2 in the extracellular space and thereby preventing its cellular entry. Indeed, they point out that there are clinical trials of angiotensin 2 receptor blockers that target the former pathway, and clinical trials of human recombinant soluble ACE2 that target the latter pathway. Boss et al. Are to be commended for their rigorous transcriptomic analysis of hypertrophic cardiomyopathy, which uncovered marked upregulation of ACE2 in hypertrophic cardiomyopathy, a serendipitous scientific finding with an uncanny connection to the current COVID-19 crisis. Our second highlight is a special article entitled Obesity and Outcomes in COVID-19, When an Epidemic and Pandemic Collide. This article is authored by Dr. Fabian Sanchez Gumar from the University of Valencia and Incleva Biomedical Research Institute in Valencia, Spain, and numerous colleagues from other centers worldwide. Obesity is among the risk factors associated with poor outcomes in COVID-19. This association is consistently recognized in the current COVID-19 literature, and in this disease there appears to be no obesity paradox, the phenomenon whereby obese as compared with lean individuals may exhibit better short and medium-term outcomes in certain cardiovascular and other diseases. Obesity generally predisposes to a number of diseases that themselves are associated with poor outcomes in COVID-19, including type 2 diabetes mellitus, hypertension, atherosclerosis, atrial fibrillation, chronic pulmonary disease, and chronic kidney disease. In the current issue of Mayo Clinic Proceedings, Sanchez-Gomar et al. Discussed the multiple pathogenetic mechanisms whereby obesity worsened outcomes in COVID-19. Adipose tissue possesses relatively high expression of the cell membrane receptor ACE2, which enables cellular entry and attendant replication of SARS-CoV-2 in adipocytes. Adipose tissue, especially in obese individuals, may thus serve as a repository for the virus from which viral spread may occur. Adipose tissue possesses the major components of the renin-angiotensin-aldosterone system and is a source of circulating angiotensin II. Angiotensin II fosters inflammation and vasoconstriction-dependent ventilation-pulmonary mismatch in the lungs, thereby exacerbating the pulmonary complications that may occur in COVID-19. Adipose tissue is a source of pro-inflammatory cytokines, including IL-6, and the systemic delivery of such cytokines to the lungs may drive acute lung injury in COVID-19. Adipose tissue promotes a prothrombotic state because of the prothrombotic adipokines it produces and plasminogen activator inhibitor mediated impairment of fibrinolytic pathways. This contributes to venous thromboembolic disease that's prone to occur and worsens outcomes in COVID-19 patients. Finally, obesity predisposes to endothelial dysfunction in systemic and regional vascular beds. The dysfunctional endothelium not only exhibits a prothrombotic phenotype, but is also a source of systemic pro-inflammatory cytokines. In addition to these pathobiologic pathways emanating from adipose tissue and converging on the lungs and other organs in COVID-19, obesity impairs the mechanics and compliance of the respiratory system, leading to impaired ventilation, decreased functional respiratory capacity, and decreased expiratory reserve volume. Sanchez-Gomar comprehensively reviews and integrates such distant and direct effects of obesity in exacerbating pulmonary and other outcomes in COVID-19, and conclude with a call for heightened vigilance when managing COVID-19 in obese patients. You can access these highlights and editor's choice articles free of charge during the entire month of July. When you visit our Mayo Clinic Proceedings website at www.mayoclinicproceedings.org, you will find links to our social media by clicking the buttons up top of the homepage to follow us on Facebook, Twitter, and YouTube. On our YouTube channel, you'll find full-length author interviews called Insights, 60 seconds video article synopses, and our Mayo Clinic Proceedings issue summary and Author Insights podcast recordings, which are available from our website on the homepage as well as through iTunes. You will also find our online-only feature, Pioneers and Legends in Medicine, which are video interviews of people who have made a strong impact in their field of study. You will also find our online-only feature, Pioneers and Legends in Medicine, which are video interviews of people who have made a strong impact in their field of study. You will see many news stories on our website that are based on articles published in Mayo Clinic Proceedings. And finally, you will see other free content and articles published online ahead of print. As always, we greatly thank you for your interest in and support of Mayo Clinic Proceedings.
We hope you found this presentation from the content of our website valuable. Our journal's mission is to promote the best interests of patients by advancing the knowledge and professionalism of the physician community. If you are interested in more information about us, our homepage is www.mayoclinicproceedings.org. There you'll find access to information for our social media content, such as additional videos on our YouTube channel or journal updates on Facebook. You can also follow us on Twitter. More information about healthcare at Mayo Clinic is available at www.mayoclinic.org. This video content is copyrighted by Mayo Foundation for Medical Education and Research.